Birds and reptiles that have a high salt intake, especially those that live in marine environments, use salt glands in order to excrete that excess salt. The salt glands are in an area above the eye, and they have a duct that goes down to the nostril. Salt gets excreted into the lumen of the duct, travels down and out. That's the location. Let's look at the structure. The structure of a salt gland has the central lumen, a central duct. Branching out from it are ducts that feed into the central duct to the nostrils. If we looked at the structure of some part of one of those ducts, what we'd see are individual epithelial cells that line it with tight junctions. And sodium and chloride move into the lumen, water follows through aquaporins, and we wind up with a salt-rich solution heading down to the central duct. What's the molecular mechanism? If we looked at one of these cells, there are tight junctions here, but those tight junctions allow sodium to move across. If we zoom in on one of these cells, this is the lumen of the duct. Basement membrane would be here, basolateral membrane, and the apical side of the cell here, the apical membrane, basolateral membrane. The sodium-potassium pump moves 3 sodiums out for 2 potassiums in, and those potassiums keep cycling around. This process takes an ATP. Because of the high sodium outside, the low sodium inside, the NKCC transporter moves 1 sodium, that's what drives this process, 1 potassium, and 2 chlorides. And this is a secondary active transporter. The process continues because sodium keeps being recycled out, keeping the sodium concentration low inside the cell, and that's necessary to keep the NKCC transporter working. The chloride moves through chloride channels. One is the cystic fibrosis chloride channel, and there's at least one more that's important. That moves chloride into the duct lumen. That creates a net slightly negative charge. Sodium is low inside the cell, but it's high outside the cell. That's what's driving this process. The space between cells is large enough for sodium to move down. At the tight junction, it is possible for sodium to cross, and so sodium moves in, attracted by this net negative charge to the chlorides. Finally, there are also aquaporins in the cells lining the lumen, and water moves passively through the cell because of the high solute concentration. This is an energy-intensive process, but it can build higher sodium and chloride concentrations inside the lumen than on the outside. The sodium will move to an area of higher and higher concentrations simply because of the attraction to the negative charge. So you can wind up with, for example, 300 milliosmoles here, and depending on the metabolic rate of the bird, you might end up with 500 or more milliosmol concentration. In other words, a constant net movement of sodium and chloride. One final piece is a countercurrent exchange mechanism. The fluid is moving sodium, chloride, and water-rich fluid. And here are capillary endothelial cells. In order to secrete salt out of the body and still be able to drink seawater, a marine animal needs to be able to concentrate salt to a higher concentration than in seawater. Seawater is about 1,000 milliosmoles, with somewhere on the order of 90% being sodium and chloride. In a marine bird or reptile, the osmolarity might be anywhere from as low as 300 to maybe more likely 350 or so. A human's around 300. So let's say they have plasma osmolarity of about 350 milliosmoles. And that encounters at some point, going in an opposite direction, it's a countercurrent exchange system, the fluid that's leaving. We know that in some marine birds and reptiles, the concentrations may be on the order of 1,200 to 1,800 milliosmoles. Clearly, the water movement must be regulated. The aquaporins must be regulated. Otherwise, they would dilute this fluid to match that of the blood. The aquaporins are allowing in enough water to keep fluid moving, but not enough to dilute this. As the blood moves, the concentration of salt is decreasing, and here we might manage an 1,100, depending on the species, to 1,700 milliosmol gradient. It's the difference between these two that matters, so we don't get quite as high a concentration. As we move to here, we are at 150, and maybe now we're at 1,000 to 1,600, again, depending on the species. We might be as low as 50 with the concentration here at something like 900 to 1,500 milliosmoles. So this high salt solution goes down into the central duct of the salt gland and is excreted. The blood, now low in salt, goes back to mix with other venous blood. So on each pass, salt is being removed from the capillaries and it's being secreted at a higher concentration than there was in the salt water that was taken in by the bird or reptile.